to as you would in real life as possible.
Alrighty, I'm going to let all our guests in. Here we go.
All right, hi everybody. Thanks for joining us. So, welcome to our session. This poster session is titled "The Bonds That Make Us." So we've got 7 exciting posters that these guys are going to share with us. And just a few reminders before I let them get started. We're here to honor the work that these undergrads have done over the years. So please, everyone be respectful of each other and be respectful of the presenters. For all the guests, thank you a lot for joining us. Just make sure that you're muted and your video is off for the duration of the session. Presenters, if you could also please remain muted unless you're presenting or then or responding to questions in the Q&A. We'll do a Q&A at the end of the session. So once you're all finished with your presentations, we'll have probably about 15 minutes for questions for everyone in the group. If anyone has questions during the course of the session, feel free to type them in the chat if you'd like, and then we'll get to them at the end. And I'm going to keep time. So the plan is for each presenter to have 5 minutes to walk through kind of to talk through some of the things on their poster. At 5 minutes, I'll give you a verbal nudge to wrap it up. And then after that, I'll introduce the next speaker and we can switch screens. And so why don't we get started with Alexandria. So Alexandria Montgomery is a student, a major in biology, and she's going to be presenting her poster titled "The Anaerobic Digestion of Wastewater Sludge in the Atmospheric Gases of Mars."
Hi, so my name's Alexandria. This is a project that I worked on last year. So let me go ahead and see if I can figure out how to share my screen here. Okay, so here we go. Alrighty, so this is the full poster. Really what I was looking at was how can we utilize a type of bioreactor that's commonly used on Earth for long-term space missions. So we have a lot of proposed long-term missions to places like Mars and Titan, and we have all these other ideas, but currently we're limited by the cost of these sort of missions and the lack of technology. So what this project is really looking at is, can we utilize methane-producing bacteria that's naturally occurring in waste for the production of energy? So right now, when we have missions anywhere, we have to collect all of our waste, whether it be by food or anything else, and we have to dry it and ship it back to Earth. And that's really expensive when it comes to things like rocket fuel and just storage space in general. So what I was looking at is, can we take all of our waste, can we put it into a bioreactor and then use the bacteria and the gases that it's producing to supplement solar panels and other things we have going on? So it's kind of trying to tackle 2 problems at once. The way that I was able to try and tackle this sort of question was in the environmental engineering lab at Oregon State. Down here in the corner of my poster, I have sort of a diagram of the methods that I was using, and it's a little bit small, so you're gonna have to bear with me, but basically what I was doing is I was setting up media bottles with wastewater sludge inside each bottle. And then I had 2 groups. I had my control group and I had my experimental group. So my control group just had the wastewater and it was sparged with nitrogen gas. So all of that headspace that was our usual atmosphere was replaced with just nitrogen. So this bacteria does really poorly in the presence of oxygen. So that's why we did it that way. That's its best conditions. And for the experimental group, what happened was it was sparged with the gases that are found at the surface of Mars. So all of those ratios I was able to get mixed at a welding supply company, and then that was filtered through the experimental group. So over time, the bacteria in these bottles was producing gas. And as you can see in that first part of the diagram, it's going to build up pressure in the headspace. And the way that I was able to then measure the pressure and the volume of gas produced is just by taking a gas syringe and by plunging it through the rubber top of the bottle, the pressure produced was able to push up on the syringe. And when it reached equilibrium, that's how you know the exact volume produced. So then in step 3 there, you're taking the mixed gas and pushing it into a bottle of sodium hydroxide. And this is because mixed gas is containing carbon dioxide and methane. And so by shaking it up in a bottle of sodium hydroxide, you're able to remove all that carbon dioxide, because what we're really interested in is that methane. That's what will produce the energy that we're after. And after that, we use the same methods as in step 2 to measure the volume of gas. And that's how I was figuring out how much methane was being produced over time. And so here in the middle, I have all of my data. So one of these graphs I forgot to include in my oral presentation that I submitted a while ago. So that's this one right here. But what you can see is some really interesting results. So by the end of 15 days, these were monitored 3 times a week over the span of 15 days. By the end of that 15 days, there was about an equal amount of biogas produced in each system, but the methane content in the Mars group was quite a bit lower. So though it was quite a bit lower, they still produced about 50% of the methane found in the control group. I mean, honestly, this is pretty cool. The gases of Mars's atmosphere are pretty harsh. We have not just carbon dioxide, but then also there's a little bit of oxygen and carbon monoxide and argon, and there's all these other things that are likely to impact the metabolism of the bacteria present. And so what we can see from this is that while the gases did impact the bacteria and cause it to produce less methane, it was still something that was able to be produced. And so it's looking like from the experiment so far, this might be a viable option. These bioreactors are used in dairies a lot already. And if we can just set up the same sort of system where we want to do a long-term mission, I think this is a great way to try and supplement the energy that we're using. Yeah, so that's pretty much start to finish what I was doing. There are a lot of future directions I'd like to go with this, but for now, this is kind of where we're at.
Great, thank you. That's perfect timing. So our next presenter, if you want to stop sharing your screen and then we can switch. Daria Wonderlick, major in biochemistry, and she's going to present her poster titled Ensembles Link RNA Thermodynamics. Ensembles link RNA thermodynamics and molecular evolution.
Great. Is my screen sharing? Okay. Yeah, so as you said, my name is Daria, and I'm a biochemistry major in the HARMS lab. And I'm excited to share my research into how an ensemble view of RNA molecules might help us better understand how their sequence relates to function. So a major goal for biochemists is the ability to design proteins and RNA with new functions for use as therapeutics or industrial catalysts. And this requires a deep understanding of how the sequence of the molecule determines its structure and function. So up here, I'm showing this relationship for the RNA molecule that I study called the adenine ribose switch. So its sequence specifies a structure that can bind a small molecule called adenine, which allows it to carry out its function of switching on a gene. And so to design a new function, it's helpful to know which sequence changes or mutations to make. Now, predicting how a single mutation produces a functional change is relatively easy, but the challenge is that most significant functional changes require multiple mutations. And so in the simplest case, mutations are independent of each other and their effects on the observed function sum together. So for example, as I'm showing here, two mutations to the adenine ribose switch that individually weaken adenine binding are even worse when combined, as I'm showing with this darker shade of red. But oftentimes mutations interact with each other, and we can usually predict that mutations that are directly contacting one another in the structure will interact. So perhaps that would result in stronger adenine binding than we would have shown than we would have expected if the mutations were independent, as shown by this light green versus the expected dark red. So to account for the difference between expectation and observation, we have to add an interaction term, which we call epistasis. And unfortunately, the origin of many mutational interactions remains unclear. So maybe two mutations in completely unrelated parts of the structure combine to produce much, much weaker binding than we would have expected from their individual effects, as I'm showing with this extremely dark red compared to the expected medium red. So the good news is that theoretical work in the Harms lab points to a potential mechanism for these mysterious interactions. The work suggests that the standard paradigm of sequence specifying a single structure might be too simplistic. Many proteins and RNA adopt a collection or ensemble of interchanging structures, which we call conformations. And the function of the molecule is now determined by the ratio of the functional conformation relative to the rest. So this ensemble view might explain mutational interactions because it considers that mutations might have different effects on each structure in the ensemble. And this can then redistribute the ratios of each conformation. So therefore, mutations may be interacting by differentially redistributing the ensemble. So theoretically, this mechanism of mutational interaction, which we call ensemble epistasis, can arise anytime a molecule populates at least 3 conformations and mutations affect these conformations differently. So my question for my project is whether ensemble epistasis is relevant for real RNA molecules. And I specifically picked the adenine ribose switch to be my model system because it has a really simple structure, chemistry, and 4-structure ensemble. And I can track the effects of mutations on its ability to bind fluorescent adenine because the fluorescence diminishes upon binding. And further, I can change the size of the ensemble by adding magnesium to selectively stabilize only 2 of the states. So if the ensemble is generating mutational interaction in this system, I expect that reducing the size of the ensemble will reduce these interactions. So to look for ensemble epistasis, I first have to detect the effects of mutations on adenine binding. And so I do this by measuring how fluorescence changes as a function of magnesium concentration. So at high levels of magnesium, there is a larger ratio of the conformation that's capable of binding adenine. And so this means the fluorescence is going to decrease, and different mutations alter this rate of fluorescence quenching. And then next, to calculate how the mutation redistributes the ensemble, I use the fraction of bound adenine from the fluorescence measurements as an indirect way to measure the ratio of the adenine bound conformation. And so fitting this data to a thermodynamic model of the ensemble allows me to determine the ratios of the remaining conformations, which I'm showing here in their associated colors. And then finally, I can analyze how the mutations redistribute the ensemble when they combine. So then from these conformation ratio plots, I can calculate the amount of ensemble diversity with a quantity called Shannon entropy, which I'm showing here in orange. And mutations change the magnesium level at which the ensemble exhibits its maximum diversity.
You're at 5 minutes, Daria.
Okay, so my preliminary results from the 2 mutations I've measured suggest that the RNA system does exhibit ensemble epistasis. So if the ensemble is the mechanism of mutational interaction, then epistasis should be highest when the ensemble is most diverse. And this correlation is exactly what I see when I overlay the epistasis between the 2 mutations in blue with the ensemble diversity in orange. So I'll wrap it up there. Thank you.
Great, thanks, Daria. So our next presenter is Stacey Andreeva. She's a chemistry major, and she's going to present her poster titled metal-ligand bond dynamics and metal-organic frameworks confirmed by variable temperature vibrational spectroscopy. Go ahead, Stacey.
Thank you. So my name is Stacey, and I'm a sophomore majoring in chemistry. And at the symposium, I'm presenting the work I've done this year in the Brozark lab. And so to generalize, the projects in the lab are centered around studying interesting properties of soft materials like metal organic frameworks, colloidal nanocrystals, ionic liquids, as well as condensed lattice materials using physical and organic methods. So my project revolved around studying dynamics in porous MOF materials and trying to establish a fundamental impact of dynamic bonding on the behavior of this material, such as determining what's the relationship between the labile bonding and conductivity that could enable self-healing mechanism in these materials. So you might ask, what's so special about MOFs? They're crystalline microporous materials noted for their enormous structural diversity and versatility. And interestingly, they emerge from dynamical bonding formation process dictated by dynamic bonds, which are sensitive to induced stress. And MOF formation depends on the assembly of two building blocks, the metal ions or metal clusters. Like in this case, we have a metal cluster of Mil-125, one of the representative examples, as well as organic ligands, like terephthalic acid shown here. And the formation process is dynamic and is actually a common phenomenon in molecular material systems because erroneous bond formation can be corrected through reversal until the material settles into the most thermodynamically favorable crystalline phase. So given the vast potential of MOFs, gas storage, separation, sensing, catalysis, and drug delivery applications, detailed knowledge of the mechanistic behavior in MOFs is highly desirable. But there is a major lack of understanding about phase transitions and the mechanism of phase transitions in MOF materials. And so uncovering this mystery could potentially be very helpful and is a fascinating platform to explore and establish structure property relationships, and very useful in the fields ranging from medicine to industry. And interestingly, from combination of metals and linkers, many well-defined structural possibilities arise, which also affect the material properties, like tunability, porosity, catalytic activity, structural rigidity. And so in our study, we were looking at the metal linker bonding in MOFs. And we observed that it is more labile than it was previously believed, and was the origin for other studies as well. And we derived this inspiration from other studies that describe the fascinating phenomena of MOFs that undergo through post-synthetic modification like cation or ligand exchange, as well as phonon softening, induced phase transitions, and sometimes even electronic transitions, and temperature-induced metal-ligand decomplexation in self-healing polymers, which was explored by Raman vibrational spectroscopy. Also, molecular dynamic simulations showed the dynamic dissociation and dissociation process between carboxylate and the metal. Therefore, we came to the conclusion that rigid bonding cannot explain MOF behavior. So we propose that this behavior could be visualized by observing the association and dissociation between the metal carboxylate and the carboxylate, I mean this part, and by infrared diffuse reflectance spectroscopy. So we focused on observing changes in the carboxylate driven by the temperature, and due to the weakness of these bonds, we can see here in the model section that increasing the number of species, that increasing the temperature would also increase the population of the state of unbound species. And so variable temperature vibrational spectroscopy would therefore show the lowering in energy. So the main goal was to identify structural changes within the system caused by applying stress onto the system in the form of heat, and more importantly, establish a quantitative model for this phenomenon, which we call the equilibrium model and just showed here. This model derives from the equation that relates equilibrium and temperature, and we assume A to be a completely associated state, which is more populated at lower temperatures, and the higher in temperature you go, the more you populate the dissociated state B.
You're at 5 minutes, Stacey.
Oh, sorry. I'll just jump to the results. So our representative system was copper BTC, and we showed that at lower temperature, we observed the asymmetric carboxylate here, and with higher temperature, it shifts to lower in energy, almost by 6 wave numbers, if we can zoom in here. And what else can I say? This could be an important work to show how we could tune properties of metal organic frameworks and achieve control of structure property relationships. Right. I'll end it here.
Awesome. Thank you, Stacey.
Thank you.
So our next presenter is Bryson Ricamona, and he's a biology major, and he's going to present his poster titled Polycomb Repressive Complex 2 Ensures Robust Skeletal Growth and Patterning During Zebrafish Fin Regeneration. Bryson, if you want to share your screen.
Hello, everyone. As mentioned, my name is Bryson Tyler Ricamona, and my project investigates polychrome repressive complex 2 ensuring robust skeletal growth and patterning during zebrafish fin regeneration. So as we all know, upon injury, most mammalian tissues will result in the formation of scar tissue. But one other mechanism that we study in the San Kunis lab is through regeneration. And we use zebrafish caudal fins as a model to understand regenerative mechanisms. Here you can see in this figure that upon amputation, zebrafish caudal fins will robustly regenerate. Upon amputation, differentiated cells at the site of amputation will de-differentiate into a more progenitor-like state. A subset of these progenitor-like cells can be characterized by this Stock-positive niche. And as regeneration proceeds, this Stock-positive niche, as well as Wnt signaling, will gradually decrease due to redifferentiation of these progenitor-like cells and reestablishment of the cell population that was lost upon amputation. One way that various cell states are thought to be maintained is through trimethylation of histone H3 lysine 27, referred to as H3k27 trimethylation, which is done through polychrome repressive complex 2 and its catalytic subunits of EZH1 and EZH2. We can see here in this diagram that upon covalently binding methyl groups to histone H3 lysine 27, histones will go from this open conformation to this closed conformation, which is thought to result in gene repression and therefore maintain cell state. We can see here that upon mutagenesis of EZH1 and EZH2 alleles, specifically EZH2, we see a significant decrease in H3k27 trimethylation levels. And zebrafish, though it's really hard to see, zebrafish tend to display underdeveloped cranial, EZH2 mutant zebrafish tend to display underdeveloped craniofacial dermal bones like the operculum and shortened BSR3 lengths. It's also noted that EZH2 mutants do not survive past 14 days post-fertilization. So one way around that to get zebrafish to survive into adulthood is that we found an EZH2 hypomorph mutation made before upstream of the WD binding domain, which allowed fish to survive into adulthood and would further be used in regeneration studies. One phenotype that we observed with the caudal fin of these EZH2 hypomorph mutants is that they displayed different overall shapes than the defined V shape that we're used to seeing in wild type fish. And when quantifying the ray lengths, I observed that the medial raise of the EZH2 mutant is proportionally similar to that of the lateral raise, and this overall shape tends to be conserved during regeneration. So this suggests that EZH2 ensures proper skeletal outgrowth in the caudal fin during development. So my specific question focused on how is PRC2 acting to ensure skeletal patterning during regeneration. Preliminary studies have shown that EZH2 is upregulated in regenerating tissue, and I also observed that H3k27 Trimethylation levels were highest at the distal tip at early time points in regeneration, like 4 and 7 days post amputation, would suggest that PRC2 is acting to maintain these progenitor-like populations at the distal tip. Looking at wide field microscopy images, I noticed that skeletal patterning defects tended to occur as early as 5 days post-amputation in EZH1 mutant backgrounds when compared to EZH2 controls. Looking at fully regenerated fins, we can really see the extent to the skeletal patterning defects where EZH1 mutants possess fusions of adjacent rays, as well as the formation of large bony plates. And we did not really see this in EZH2 mutants and wild type controls. We did a secondary amputation, and the skeletal patterning defects were conserved upon this second round of regeneration. So my proposed model is that upon the lack of PRC2, these differentiated cells will either go back into this multipotent progenitor-like state, as seen, which could be seen during development, and this multipotent progenitor can go on to various cell fate pathways, or that differentiated cells will de-differentiate into other cell fate pathways, which result in the excess of different cell types in the regenerating tissue. In conclusion, overall, my results suggest that PRC2 acts to maintain various cell states during regeneration. Thank you.
Awesome, thank you, Bryson. So our next presenter is, I'm sorry if I messed this up, but Ariana Entezari-Schweiger, hopefully that's right. And she's a major in human physiology and is going to present her research on her poster titled Stem Cell Research.
Yeah, thank you. So I'm a part of the Dryer lab, but I decided to go with researching my literature synthesis that I did for a class. And it's on stem cells and basically what they are and how they interact. So basically stem cells have already begun to cure people with seemingly incurable diseases such as Alzheimer's, spinal cord injuries, things like that. These cells are already responsible for curing a list of problems and are producing insulin for those with pancreatic beta cell mutations. Stem cells have progressed in the last 40 years or so, but there's controversy between whether or not it's moral or immoral and whether we should continue doing them. And basically the stem cells differentiate once you add them into the muscle type and divide into different cells, basically replacing the already damaged cell, which is very regenerative. Nerve cells can be tested for drugs that may help repair neurons and for nerve diseases like cerebral palsy and carpal tunnel syndrome. The cons are very valid with exactly how far are we going to go with this kind of treatment. Are we going to start changing things such as eye color, IQ that could happen, gender, things like that? Where will we stop the technology on creating different cell types? Many people say that this is basically playing God as a scientist in genetic engineering. But I think the pros kind of stand out with them differentiating and replacing cell types that are damaged or lost due to aging and disease. This process is deemed regenerative or reparative medicine. We could continually replace all cells in our body we could potentially extend the human lifespace. We can't do research without funding though, which ties into the politics surrounding it. There currently aren't any federal laws that are banning stem cell research. However, there are a lot of restrictions on it, and there's a lack of funding, it's hard to continue on with your research, which is slowing down the production of the information that we can gather with less financial support. And if we expect to make true strides within this field, we need funding in order to make this happen. Stem cells are very controversial, which I brought up. The use of embryos and the cost of potential therapies. The development of regenerative therapy to continue the research necessary to inhibit things such as tumor growth, post-implantation. Stem cell research is a growing field. The potential benefits of stem cells include therapies of neurodegenerative diseases, arthritis, cystic fibrosis, and the chance to test new drugs for safety. Ultimately, stem cells are very evolutionary in the medical field. I kind of have a picture, a few right here, but you can't really see them very well. But basically, they take an embryo, which is in its 5 to 10 day stage, and they extract it. So it's still undifferentiated, and then they place it into the tissue. They can either make it in their cell or in their lab and differentiate it that way, or they can just use the undifferentiated from embryos. Yeah, that was pretty much what I did. The conclusions are, well, what I concluded was it's a great revolutionary medicine. Yeah, thank you.
Great, thank you, Ariana. So our next presenter is Nathan Stovall, and he's a major in chemistry and is going to present his poster titled Ultra-Thin Iridium Oxide Catalyst on a Conductive Support for the Oxygen Evolution Reaction in Acid.
Okay, now I have to figure out how to unshare. There we go.
Very cool. Yeah, I'm Nathan Stovall, and I am a sophomore chemistry major at the University of Oregon. And I have had the privilege of working in the Betcher lab under the guidance of my mentors, PhD candidate Raina Kravina and Dr. Shannon Betcher. And we've worked to develop and characterize an ultra-thin iridium oxide catalyst on a conductive support for the oxygen evolution reaction in acid. But why do we care about the oxygen evolution reaction in acid? Well, as you guys probably know, there's a growing demand for clean energy throughout the world, and one of the very promising technologies to meet these needs is using hydrogen gas as fuel. Hydrogen gas can be produced via water electrolysis or water splitting in a water electrolyzer, where you evolve oxygen at the anode and hydrogen at the cathode. More specifically, the state-of-the-art proton exchange membrane, or PEM, electrolyzer has shown a high degree of promise, but is limited by the slow kinetics of OER and the harsh acidic operating conditions. Thus far, the only known catalyst to produce high activities and high stability under the harsh operating acidic conditions of PEM electrolysis is iridium, but due to its low abundance and high cost, it hasn't been feasible for the widespread implementation of this technology. So to this end, our goal has been to develop and characterize a reduced loading iridium catalyst supported on a cheap acid-stable metal oxide support while maintaining the high activities of a bulk iridium catalyst. This led to our development of a novel synthetic method in which we can soak antimony doped tin oxide, or ATO, nanoparticles in an organic solvent with this iridium dimer we call iridium cod. When you apply mild heat and in a nitrogen atmosphere, you can adhere iridium to the surface of this ATO. We wanted to look into what mechanisms and how these were binding to each other. So first we did a UV Vis experiment in which, as the reaction ran, we tracked the absorbance of our iridium cod dimer over time. And after about 2.5 hours, as you can see, this absorbance plateaued in both toluene and acetonitrile. This suggests that our reaction is surface limited, likely by the surface hydroxyl groups of ATO. We did a further investigation using nuclear magnetic resonance spectroscopy, where we used a model metal oxide triphenylsilanol that is suitable for NMR, and we tracked this hydroxyl hydrogen before and after reacting with the iridium dimer. So before, we can see this peak here corresponds to this hydroxyl hydrogen. And then in the middle, we have the iridium cod NMR, which has no hydrogen peak, as you'd expect. And then after adhering the iridium to the triphenylsilanol, we see a slight shift in this hydrogen, but we do not see its disappearance. This suggests that quite likely our iridium dimer is adhering to the surface of our metal oxide instead of a direct bond, possibly through the partial positive hydrogen of the metal oxide hydroxyl group and the partial negative chlorines in our iridium dimer. And sorry, move this thing. And next, we wanted to develop a calibration curve that allows us to track the loading of our catalyst on the surface of our electrodes before running electrochemical measurements. And to do this, we were able to use X-ray fluorescence and relate the peak area of X-ray fluorescence data to a known mass of iridium that we were able to calculate using a quartz crystal microbalance on a quartz crystal. And as you see, you get a linear relationship, which then, as we build our electrodes, will allow us to determine the mass loading on that electrode and therefore compare catalysts between each other as a function of their mass. This leads us to where we tested their electrochemical performance, where we ran what's called a cyclic voltammetry experiment, where you sweep between a known potential range back and forth and measure the current response. In this case, we're denoting it mass activity, which is simply current per unit mass. And what we're looking for is an earlier current response, as you see in the screen in blue trace, both of which are our novel catalysts that we've developed, and we're comparing it to iridium oxide nanoparticles, which, as you can see, are significantly less active. This is likely due to the fact that bulk iridium nanoparticles then cover each other's active sites, making the overall catalyst less active, as opposed to our catalyst, which is very thin, where most of the iridium active sites should be present to the solution, allowing for those iridium active sites to facilitate the oxygen evolution reaction. We also wanted to test the stability of our catalyst through many cycles of cyclic voltammetry, as that has been the problem reported in the literature, that these ultra-thin catalysts are not very stable. And we found something similar, where cycle 1, you see here, it was very active, but over time, we saw a later and later onset of current, which denotes later and later onset of our oxygen evolution reaction.
You're at 5 minutes, Nathan.
Cool. This likely could be due to the weak binding interactions of our iridium dimer to the surface hydroxyl groups. And lastly, I just want to say thank you to the Betcher Lab for the tremendous opportunity to do research in your group and have the opportunity to work with Raina and her tremendous mentorship. Thank you.
Great, thanks, Nathan. So our final presentation is Laura Reich, and she's a biology major and is going to present her poster titled Utilizing Behavioral to Study Gap Junction Channels in Developing Zebrafish. Go ahead, Laura.
Right. Hello, everyone. My name is Laura, and my research is all about gap junction channels and identifying connexins in zebrafish. And so to orient you to my research, I first want to start out by noting that the nervous system and the musculature coordinate activity to elicit motor behaviors like walking and running. And both of these systems utilize a number of subcellular structures in order to do this. One of these structures is known as gap junction channels. Gap junction channels connect two adjacent cells together and allow for the direct flow of small molecules. It's also important to note for us that gap junction channels are composed of proteins known as connexins. Now, the connexin family is large and complex. Humans have about 20 connexin encoding genes, and zebrafish have about twice as many, around 40. Despite this, we don't know how individual connexins contribute to behaviors. And so to sort of tackle this question, we decided to use embryonic zebrafish as a model organism because these zebrafish elicit a motor behavior from 17 to 24 hours post-fertilization known as spontaneous coiling. And spontaneous coiling is pretty simple. The zebrafish just flicks its tail back and forth, and we know that this behavior requires gap junction channels. So I'll try to zoom in here. The coiling circuit right here is the circuit that gives rise to the spontaneous coiling. And all of these red boxes are showing where gap junction channels are located, either in the neurons or in the corresponding musculature and slow muscle fibers. So we know there's these gap junction channels here, but we don't know the individual connexins comprising them. And so my big question was to use this model of embryonic zebrafish and spontaneous coiling to then investigate what these individual connexins are that comprise these gap junction channels in the coiling circuit. As my first step at doing this, I first wanted to develop an automated behavioral tracking system to screen for connexin mutants. If we do this, the idea is that if we find a connexin mutant that has defects in proper spontaneous coiling, that would indicate to us that that specific connexin is within the coiling circuit. And we decided we wanted to validate the accuracy of this system with wild-type zebrafish as well as connexin 46.8 mutant zebrafish. This specific connexin is already known to reside in the slow muscle fibers of the coiling circuit at 1 day post-fertilization. And we already know that mutants for this connexin have a coiling defect. So it serves as a really great tool to use to validate a system. And we decided to use Deep Lab Cut, or DLC, as our automated behavioral tracking system. It's essentially a neural network that can learn to recognize patterns in an organism as it executes a behavior. And we first have to train DLC in order to do this and in order to track coiling. So what we did is we first captured 150 frames of wild-type coiling. Then we manually defined points on these frames. We defined 6 different points. So you can see that in the image I have right here. We had a point on the top of the head, 2 sides of the yolk, and the start, middle, and end of tail. And these are the points that we're training DLC to be able to notice and track for itself. Once we defined all of these points, we then trained the DLC network with the UO Tilapia supercomputer. From this point, the big test is to provide our network with a novel video of spontaneous coiling that the DLC network has never seen before. And we want to see if DLC can place these points on the organism for itself and properly track coiling. So that's what we did. And we found that yes, DLC can properly track coiling behavior, not only in wild-type zebrafish, but also in these connexin 46.8 mutant zebrafish. And so I have these trajectory plots in front of you. Each trajectory plot right here is showing the movement of these points for that zebrafish in the course of a 30-second video. And so if we look just at these wild-type trajectory plots, each one's taken at a different time point in development, we can see that the orange dots and the dark red dots expand and have a wider range over time. That's specifically the middle of tail point and the end of tail point. So we see that this wild type has a pretty wide range of motion once you get to 20 hours post-fertilization. This is not the case when we're looking at the mutant zebrafish. It has a much more narrower range of motion. Thus, through the DLC data alone, we're able to validate that the system is able to track coiling behavior, and it's able to differentiate between coiling phenotypes and see a weaker coiling defect or a weaker coiling phenotype in these mutants. The next part of my research I would like to talk to you about is that my next step was to begin to identify some of the connexins that might be at the coiling circuit neurons at 1 day post-fertilization. The idea is that if we find candidate connexins in these coiling circuit neurons, we'll be able to make homozygous mutants for them to then run them through this DLC network that we've been able to create and generate. So for this antibody staining, I wanted to first mark where coiling circuit neurons are. So I used OLIG2 for that. And then I used a human connexin 36 marker. And this human connexin 36 marker recognizes 4 connexin 36 fish orthologs. These 4 are right here in this corner, and they're already known to reside in neurons of fish at other time points in development, but we specifically wanted to look at 1 day post-fertilization. So you'll see my antibody staining. Cool. You'll see my antibody staining right here. The magenta is the OLIG2 coiling circuit neurons. The cyan is where the connexin 36 marker is. And so you can see this punctus staining for the connexin 36 marker right at the magenta, indicating to us that some of these connexins might be at the coiling circuit neurons. And then in terms of future directions, we want to be able to generate homozygous mutants for these connexin 36 fish orthologs, run it through our DLC network, and see if they really are required for spontaneous coiling. Thank you.
Awesome. Thank you, Laura, and thanks again to all of our presenters. Now we'll open it up for some question and answer for all of the panelists. I think we can probably start with a question from Mike Harms in the audience that was for Bryson, and then we can open it up also to the rest of the panelists. Mike, if you'd like to unmute yourself and ask your question, you're welcome to. Otherwise, I can just phrase it.
Sure, I'll read it. Thanks everybody. And just my question for you, Bryson, was how you separated out whether this factor was responsible for maintaining these cells in their regeneration lineage, or if instead it was important for entering that regeneration state.
Yeah, that's a great question, Professor Harms. And am I able to share my slide?
Sure.
Okay. So I kind of concluded that PRC2 acts to maintain cell states versus their transitions. First indicating by this result, though these results are very preliminary, it seems that H3k27 trimethylation levels are highest between 4 and 7 days post-amputation, which is well after differentiated cells start to de-differentiate, which occurs around 2 to 3 days post-amputation. And these early time points are typically associated with relatively high progenitor-like cell populations. And in terms of redifferentiation, we can see that in these wide field microscopy images that these skeletal patterning defects tend to occur relatively early, but you can see that at the distal ends of the fin, these phenotypic defects tend to correct itself. So that kind of suggests that these cells are able to redifferentiate, and that PRC2 is mainly acting in this like initial outgrowth of the fin, which is associated with relatively high progenitor cells.
Cool.
Does that answer your question?
Yeah, no, that answers it very well. I mean, you probably even said it going, but I missed it in the flow. So, but cool work. Thanks.
Thanks.
And if anyone else in the audience has questions, you're welcome to unmute yourself and chime in. While we're waiting, I'll kick off with a question, I guess, that any of the panelists are welcome to field. I feel like when the real learning happens in science, when stuff doesn't work the way you expect it to. So I'm kind of curious whether any of you can tell us about some of the challenges that you encountered in your project and how you worked through those challenges and overcame them. So if anyone who wants to chime in on that, you're welcome to.
Yeah, I can speak on that. So I can share my screen again quickly, but if you remember from my presentation, I was talking a lot about training this DLC network, and from my presentation, it might have sounded like a very seamless process. Like we found the perfect frames to capture, and then we defined the points, and then voila, it tracked it perfectly. But that really wasn't the case at all. There was a lot of trial and error in providing the DLC network with frames that would allow the DLC network to really be able to track coiling behavior. So the 150 frames of wild type coiling we ended up using from 4 different videos of wild type coiling that had these wild type zebrafish at different points in development. And some of them had slight changes in their orientation to ensure that when DLC is being provided a novel video, it can track that coiling behavior, even if that novel video looks a tiny bit different from something it hasn't seen before. So there was a lot of trial and error in that.
That's cool. Alexandria, actually, I'll kind of point that question towards you, because I just caught that you mentioned that you had to work with a welding supply company in setting up your research. I mean, was that kind of a trial and error and something you had to figure out from scratch?
That's actually probably the detail that came together the easiest. what I found was really cool was that all I had to do was send the welding supply company a list of the exact ratios that I wanted, and they just mixed it together there for me in their lab and just sent it over in a big canister. So I had a giant gas chamber that was just full of the ratios that I asked for. The really difficult part was finding bacteria to use. So you can buy samples online from biological supply companies of just methanogens, but they're often really hard to culture and grow in a good time period. And so I originally wasn't planning to use wastewater. It's sort of an unconventional source of biomass for a project, and it causes a lot of safety concerns. And so the trickiest parts were deciding to use something like this and then finding a lab that could handle that sort of risk. It took 2 years to figure out how to obtain this bacteria and how to use it. And then from there, everything was pretty straightforward.
That's pretty cool. Does anyone else want to tell us about some of the unexpected challenges they ran into? I guess also kind of a related question, you know, if you could start the project from scratch again, knowing everything you know now, you know, is there anything in particular that you do differently in your project?
I think for my project particularly, referring to that preliminary immunofluorescence staining figure, I definitely like to capture more time periods during regeneration to kind of get a more general aspect of how H3k27 trimethylation levels are affected throughout a broader sense of regeneration, as well as possibly testing more zebrafish within my experiments.
Awesome. Corinne, go ahead, Daria.
One of the hardest parts with my project is just working with RNA in general. It's actually like super sensitive to work with, and you have to have like a really like sterile lab environment. And so I've actually spent almost a year and a half just trying to like make enough of the RNA to be able to get measurements. And so one of the like big ways I was able to like up my yield was collaborating with a lab that actually specializes in RNA work on campus. And so if I had made that collaboration like a year earlier, I probably would have had results a lot sooner than I did, but I'm happy that I was able to make that connection.
Right, and sometimes those, you know, going through the process of searching for that is instructive in itself, right?
Definitely.
Corinne Stankunas in the audience has another question for Alexandria. He's wondering if you have any plans to use atypical or modified bacterial strains. So, you know, for example, screening for, you know, evolved strains that might produce more methane.
So I think this is a really interesting avenue to take this project in the future. What I was looking at is how can we utilize the waste that we're already going to be producing on these missions. So right now we have to save everything and send it back, but I'm looking at, can we just use what we already have? So I think creating a more efficient system would make this an even more worthwhile goal, but it would be difficult to look for these evolved bacterial strains, because if you found a strain that did better, that's another resource you have to ship up for these missions. So by starting with the baseline of what you already have, I think that's a great starting point. And if there is a way that we can find to practically store and ship up maybe some bacterial strains that can reproduce and spread throughout the system, that is a great way to try and improve the efficiency and create a greater output of energy.
Great. Thanks, Alexandria. Stephanie Stovall in the audience has a question for Nathan. So how does your lab plan to address the instability of this catalyst you're proposing?
There are a couple ways. One way that we could do it is we could test different reaction conditions. to possibly facilitate stronger binding interactions with the metal oxide. If you remember, we thought it was likely only adhering through electrostatic interactions, but if we could get a stronger bond, that would quite obviously make it a lot more stable. And we could also investigate the use of oxidized and stable ligands on our catalysts, which the catalysts we've built now, the ligands under the oxidizing conditions of OER, they'll dissolve and they'll fall off, which was on purpose. But potentially the use of ligands that are stable under those conditions could make it more stable as a whole.
That's awesome. Thanks for your question. We should probably, Erin, I think we should probably wrap it up. They want us to be very strict about ending on time so that the next session can proceed. Is that right, Erin?
Yes.
Okay, but thanks again to all of the presenters and for the audience for asking great questions. You guys have done really, really impressive work. So please keep it up and we'll see you again.
Congratulations. It was great presentations.
Thank you all. Bye-bye. Thanks for your help, Erin.
Oh, totally. You did great.
All right.
Yeah, excellent. Great questions. And yeah, everything worked very smoothly. So that was awesome.
Great.
What department do you work in?
I realized I forgot to introduce myself. I'm in the Institute of Neuroscience.
Oh, okay, great. Great. I'm in the libraries. I help write grants and do administrative stuff.
Hopefully I'll run into you when I need help writing grants.
I might need help with your topic, but yes. Well, it's nice to meet you.
Thank you so much.
All right, see you around.